This is very basic biochemistry for biology students. It will help you tremendously later on because we'll be talking about the phospholipids in the plasma membrane or steroid hormones. That's why you need to have a basics in biochemistry. Very brief, I promise. Macromolecules are big molecules, and most biological molecules are big. Okay? They usually include carbon, they have hydrogen, they have oxygen, sometimes nitrogen and phosphorus and sulfur. Rare, sometimes you can add some other elements, but these are the main atoms that form biological macromolecules. Like I said, they're very big molecules. Most of them are made up of small molecules that are joined together, kind of like a necklace made up of beads. Okay? So the building blocks are called monomers. That'll be the bead. A polymer is formed by combining many monomers. That's when you get your necklace. Okay. There's 4 major types of macromolecules. You have carbohydrates, lipids, proteins, and nucleic acids, and we're going to have a very quick look at all of them. Carbohydrates refers to sugars and starches, so they're a major source of fuel for the cells. Not only major, but I say it's immediate. If you eat sugar, it doesn't take very long. You need to pick me up, get a candy bar, right away, you feel better. There's 3 major sizes. Monosaccharides is just a monomer. So those are the simple sugars. And yes, it's a good idea to know the names of the most important ones. So glucose, that's the blood sugar. Fructose, that's the sugar found in fruit. Galactose, all of those are monomers. If you have 2 monomers, they're called disaccharides. And this is a dehydration reaction, meaning that you have 2 monosaccharides. When they come together, they lose a water molecule, and now you form a disaccharide. That's why it's called dehydration reaction. And examples are sucrose, that's table sugar, lactose, that's the sugar defined in milk, maltose. If you have many monosaccharides, all of them linked again by dehydration, you form a polysaccharide, and the most common ones are starch, glycogen, and cellulose. If you notice glucose, galactose, and fructose, they all have the same formula, but the atoms are organized different. Looking here, for instance, you have an OH, and here you have an oxygen. But here you have an oxygen, and here you have an OH. If you count them, they all are C6H12O6, but the atoms are distributed different. Sometimes the locations to the right or to the left. You see this OH in here? That matters, whether to the right or to the left in the molecule. Those are called isomers or isomeric molecules. Okay? You do not need to know the formulas. That would be silly. These are just some examples. Starch is produced by plants. Glycogen is produced by animals. Cellulose is produced by plants. Don't worry about carrying right now. I want to talk about these 3 because you say, well, they're all made of glucose. They're polymers of glucose. But they're very different. If you compare starch and cellulose, yes, you have one glucose after another, but in cellulose, one of the glucose is upside down. You see the top of this one is at the bottom. So every other sugar is upside down. Why is that important? The chemical bond is different. The bond in the starch is very easy to break, so it's a very good source of energy. If you eat spaghetti, that's starch, you can break down one at a time and get the glucose, and you get the energy real quickly. Cellulose, because it has that add bond, every other bond is a different bond, you don't have the enzymes to break it down. No animal has it. Actually, only bacteria can break it down. So cellulose is mostly a structural carbohydrate that keeps plants straight up. Okay? Now you could say, Well, how about cows? Cows eat grass. Grass is made of cellulose, yes, but cows have bacteria in their gut that can break cellulose. A cow cannot break cellulose. My granddaddy was a dairy farmer. I still remember one summer I got to the farm. I like to go 1 month every summer. And there was this cow that looked real down, and I asked, what's wrong with her? I don't know. She doesn't seem okay. Well, have you called the vet? No. He doesn't know anything. He's a college boy. That's silly. It's okay. We have some antibiotic, we have penicillin. We are just going to give her a shot. The next day the cow didn't look any better. We'll give her another shot. And they kept doing that until the cow died. When the cow died, then they called the vet. Isn't that crazy? So the vet comes and says, I don't know what the problem is. Looking at the cow, you'd imagine she had starved to death, but I can see that there is hay and there is grain and there's all sorts of food around it. And I just had to jump and say, I know what killed the cow. And everybody looked at me like, who are you? And I explained to the vet, they've been giving her antibiotic every day for a week. That killed the flora, that killed the bacteria that digested the cellulose. So the cow was eating, but couldn't digest the cellulose. It went right through her, and she starved to death. Now, I don't know what the original problem is, because there was no way then to figure why she was down to begin with, but they killed her. They starved her to death because animals cannot digest cellulose. Glycogen is made by animals. It's a very good source of energy because it's just like a starch. The only difference is it has branches. It's even better because instead of breaking just one glucose at a time, if you have 2 branches, you can break 2 glucoses at a time. We're not going to worry about carrying now, don't worry. The other group was lipids. Lipids are very variable. There's only one thing they have in common. They're insoluble in water. They're very hydrophobic. They have many functions because they're so variable. So they can be used to store energy, as insulation, remember the polar bear? Waterproofing, you see this oil? The fur doesn't get wet. The water runs off the fur because of the lipids in there. They're the building blocks of many hormones. They're very important in the plasma membrane of cells. There's 3 major groups of lipids: triglycerides, phospholipids, and steroids. And we're going to just say a couple of words from each one. I think I just put in the wrong, but this is an example of a triglyceride. You have a molecule of glycerol and 3 fatty acids. These long things are fatty acids. This is an steroid. You will go back to this again when we describe them, but you see the 4 rings characteristic, and a phospholipid. You have also the 3-carbon glycerol has 2 fatty acids and a phosphate. Okay, so maybe this wasn't the best place to put it. But we said triglycerides has 3 fatty acids and attached to a glycerol that has 3 carbons. Now, fatty acids can be saturated or unsaturated. The saturated have all carbon, single bond, carbon bonds. Unsaturated have some double bonds. And this is very important because you see the saturated, they're almost a straight line. So you can pack many saturated fatty acids. Basically, saturated fats are likely to cause cardiovascular disease. The pack, they get hard, but the unsaturated, whenever you have a double bond, it has a bend. So you cannot pack it quite as tightly. It's like trying to have a bag of potato chips or a bag of Pringles. The Pringles are going to fit on each other real good, and you can pack them very tightly. The potato chips are going to leave lots of empty space. So saturated fatty acids are bad for you. Unsaturated, they're good. They don't promote cardiovascular disease. In fact, they prevent cardiovascular disease. Now, when you have unsaturated fatty acids, there's still a difference. Are the chains going to be on the same side of the double bond? Look at the example that you have in here. The chain is on the top, and continues from the top. So this is a cis fatty acid. But if you have a trans, and I don't know if I have a picture someplace, yes, you can see the cis, there is a curve, but with the trans, there's really no curve. This is not a very good. It continues like this, continues like that. So there's a bit of a kink, but not enough. Trans fatty acids are very, very similar to saturated, so they're just as bad for your health. Okay? When they're solid at room temperature, we call it triglycerides fats. When they're liquid, we call them oils, and definitely they're much better for you. And the main function of triglycerides is going to be energy storage, insulation, and to some degree protection, like waterproofing. Okay? Phospholipids, we said that you have two fatty acids, a phosphate, and you can go back to the drawing, and the glycerol. Now, what's interesting is that the fatty acids are very hydrophobic. The phosphate, on the other hand, is very hydrophilic. So you have a molecule that's hydrophobic on one end, hydrophilic on the other one, and that's what makes it a component of the cell membrane. Because let me represent the hydrophilic part, the phosphate as a head, and the tails are going to be the two fatty acids. By making a double layer, you put the hydrophobic parts in the center, because one of these is going to be inside the cell, the other side is outside the cell. It doesn't matter if it's inside or outside, there's a lot of water. There's water inside the cell, there's water outside the cell. So the hydrophilic ends are facing the water. The hydrophobic are hidden inside the sandwich, away from the water. And this is going to be the basic for the plasma membrane. We're going to spend a lot of time discussing the plasma membrane in another chapter. Steroids, they can be recognized for those 4 fused rings. There will be, depending on the steroid, different things hanging from there, but they all have those 4 rings. Many hormones are steroids, a number of vitamins are, bile salts necessary for digesting fat, those are steroids. The third group was the proteins. The proteins is a polymer of amino acids. So some people call them, instead of proteins, peptides. But the monomer is the amino acid. The name tells you that it has an amino group, that's an NH2, has an acid group, that's a carboxyl group. So you have here an amino acid, again you have the acid, you have the amino, there's always one hydrogen, and then there is a side chain that varies. So you have 20 different amino acids because of what is in place of the R chain. Okay, the different amino acids are linked together by peptide bonds. There's a bond between the amino of group of one and the acid group of the other one in there, and then the protein has to fold in a specific way to have a function. The folding of the protein is very, very important for the function. So you have the amino acids. When you form the peptide bonds, you have a peptide. When the peptide bond folds in such a way that is functional, then we call it a protein. Functions, very, very well. Okay? Proteins are, if I had to choose the most important molecule, probably I'd pick up proteins. They can be structural proteins. They can regulate functions in the body. They can contract, like in your muscles. They protect, think on antibodies. They transport, hemoglobin transports oxygen. They store. There's proteins that store, for instance, phosphate to be used sometimes for cellular respiration. They're a very important component of the plasma membrane. We'll see that. They're enzymes. Those are catalysts in biochemical reactions, means they speed up chemical reactions. And some hormones are proteins, so proteins can do just about anything. Okay? They can have different shapes. Some are globular, like a blob. Some are fibrous, long proteins. And we say the protein shape determines the function. If the protein loses the shape, why do they lose the shape? Well, if you change the temperature, you change the pH, if you expose it to chemicals, if you change the shape of the protein, it loses the function, and that process is called denaturation. Sometimes a protein can be renatured again. Most of the times, it's a permanent destruction. It's like when you cook an egg. You can cook an egg, but you cannot uncook it. When you cook an egg, the heat denatures the protein in the albumin of the egg, and you cannot undo it. You cannot regenerate the egg. The structure of the protein has 3 or 4 different levels. The 4th level doesn't happen in all proteins. They all have primary, secondary, and tertiary. This will be all proteins, but some have a quaternary structure. So the primary structure is the sequence of amino acids. The primary structure is going to start folding due to interactions between the peptide bonds. Basically, in the peptide bonds, you're going to form hydrogen bonds in there. It's never contiguous. There may be 2 or 3 peptide bonds in between, but it can fold as an alpha helix. You can see here an alpha helix, like a helix, or this one that's folded like a map. It's called a beta pleurisy. But now, the secondary structure is going to keep on folding. This time it's interaction between the R groups, not between the peptide bonds, but between the R groups. It can be that you have an acid group and a basic group, and they're going to attract each other. It can be forming hydrogen bonds, whatever. The protein is going to fall. And for many proteins, that's the end of it. But some proteins, you have different subunits, and they have to come together. For instance, hemoglobin has 4 subunits. It's an alpha 2, beta 2, has nothing to do with alpha helix or beta. That's how they name. There's 2 identical subunits, and 2 maybe make them a square to indicate that they're different, and all 4 have to interact to make the hemoglobin. If you have a quaternary structure, obviously you have primary, secondary, tertiary, but again, not all proteins have to have a quaternary structure. Only some of them do. Some proteins, like insulin, don't have 1 subunit, so there's no quaternary structure. The last type of molecules, and you're going to spend several chapters talking about them, so I'm just going to go very, very quickly, is the nucleic acids. 2 major types, DNA and RNA. DNA is the deoxyribonucleic acid, RNA is ribonucleic acid. The important thing about DNA is that it stores genetic information, and when an organism reproduces, it replicates the DNA to pass a copy of that information to all of the offspring. Okay? RNA, you will study the functions, there's different kinds, you'll get to that, don't worry. Both DNA and RNA are polymers of nucleotides, so sometimes they call them polynucleotides. What is a nucleotide? It's formed of a phosphate group, a pentose sugar, meaning a sugar that has 5 subunits, and there's 2 of them, ribose and deoxyribose. Obviously, ribose goes to RNA, deoxyribose to DNA, and a nitrogen-containing base. And let me show you some. I don't expect you to memorize the bases right now. Like I said, we're going to do a lot with nucleic acids. But here you can see the phosphate is very easy to identify. The sugar is always a ring with an oxygen center in there. If it has 1 OH, that would be a deoxy. But sometimes there's an OH in this other one. Whenever there's nothing in there, it means there's a hydrogen. And that one, when you have 2 of them, that would be ribose. And then there is several, let me just stop, nitrogenous bases. They can have 1 or 2 rings. We're going to see all of that, don't worry. But they have a lot of nitrogen. Most organic molecules are mostly carbon, hydrogen, some oxygen. Look how many nitrogens this one has. Okay, so when they all 3 come together, they form a nucleotide. And like I said, I'm not going to get into more because we're going to spend a lot of time talking about DNA and RNA. The only thing I want to say about DNA is that it forms a double helix. You have 2 strands. The nitrogenous bases are always in the center. The backbone is the sugar phosphate, sugar phosphate, sugar phosphate. The bases are in the center, and the form hydrogen bonds between them. A base cannot face any base. It only faces what they call the complementary base. Adenine always pairs with thymine. Cytosine always pairs with guanine. And you will learn what these are. But these will form hydrogen bonds, and that gives the two strands together. Okay? Don't worry if this is confusing, because like I said, you're going to spend a lot of time talking about DNA. So you want to work with me one-on-one? You want to have access to the tables that I've made to summarize things where you can get an overall picture or compare similar things? You want to start to prepare for an exam? There's a link to my Wise and Turing profile in the video description. You're welcome to make an appointment anytime.